Okay, welcome everyone. It is my pleasure to introduce our second keynote speaker of the conference, Dr. Orrin Devinsky. Today we will have Dr. Devinsky's keynote, followed by Dr. Jason Buss's Foundations lecture on applying the principles of evidence-based medicine to medical cannabis. That session will be in the same Zoom link immediately following the keynote lecture with a quick 5-minute bio break. To say we are delighted to have Dr. Devinsky really understates it, because in the field of medical cannabis, there is a really very wide spectrum of evidence, and it ranges from quite strong to very inconsistent and weak. And at that really strongest end of the spectrum is some of the work conducted by Dr. Devinsky in his laboratory on cannabidiol for often intractable, frequently recalcitrant pediatric seizure disorders. Dr. Devinsky is professor of neurology, neuroscience, surgery, and psychiatry at the NYU School of Medicine. His epilepsy research includes cannabidiol, but also phenome-genome correlations, precision genetics, healthful behavioral strategies, therapeutic electrical stimulation, cognitive and behavioral issues, and surgical therapy. He's written more than 500 peer-reviewed scientific papers and founded several bioscience companies. Please join me in welcoming Dr. Devinsky to the CMCR conference. Thank you so much for being here.
Thank you so much, and thanks to everyone and the conference organizers for this gracious invitation to present. So cannabis has been something I have been interested in for the past decade as an area of medical research, and I'll summarize some of my explorations in that field. So these are my disclosures. The research I did with GW was really free of any financial interest, but subsequently have become involved in several cannabis-related companies. I don't think any are relevant to this talk. So to begin at the beginning, there are two species of cannabis plants that have been used by humans for up to 10,000 or more years. The oldest that we have record of is Cannabis sativa. It comes originally from China and Chinese writings, and probably dates to about 10,000 years ago. It was used in religious ceremonies, was used to make rope and other products, and then probably not long afterwards was used medicinally as well. Cannabis indica has a shorter history as far as its written record. It comes from India, as its name implies, and this is actually the first species of cannabis that entered into England and Europe through England's relationship with India, and also in the United States in the 1800s and 1900s. We will focus today's talk on two cannabinoids, cannabidiol and tetrahydrocannabinol, THC, which I think most people are familiar with. But it is worth pointing out that there are more than 500 cannabinoids, as well as the more aromatic terpenes and flavonoids that are present in these species. So these species, like most plants, contain a large number of phytochemicals, which I like to point out are not there for Western or Eastern medicine's benefits. They are there for the plant's benefits. And many individual plants can have dozens, if not 100, different phytocannabinoids or phytonutrients. And these are all there to do something for the plant. They may prevent desiccation. They may prevent invasion by insect species. They may prevent grazing by herbivores. But they're there for the plants. They're not there for us. By coincidence, many phytochemicals are there that serve the plant and evolution, its own history, but they're also beneficial in medicine. So the two we'll focus on here are cannabidiol, or CBD. This is not psychoactive. You can take as much as you want. You may get tired, but that's essentially the only mental effect that this will have. Perhaps a little relaxed, but that would be about the max. And it works on multiple sites. I'm going to focus on a few. We think for epilepsy, the most relevant one is the GPR55 receptor, but it also works at the TRPV receptors. Among these are the ones that allow us to taste capsaicin, or the chemical in hot peppers that makes them hot. It also, CBD, acts as a stimulant of the serotonin 1A and 3A receptors, and it's likely that this effect is related to some of the anti-anxiety properties I'll review briefly. And also, CBD has a remarkable range of anti-inflammatory effects. THC, which is psychoactive, has been studied for a longer period of time, and we actually have a bit more history about CBD and its receptors in the brain. These both compounds were identified by Meshulam in Israel back in the 70s, and actually he did some trials in epilepsy going back to the late 70s and early 80s. Well, THC, as I said, is psychoactive, and it's psychoactive because it stimulates the CB1 or cannabinoid 1 receptor in the brain. It also stimulates the cannabinoid 2 receptor, which is located primarily outside the brain on immune cells, but also in microglia, which are among the brain's immune cells. This slide, which is a busy one, and I'm happy to make my slides available to anybody, so don't get caught up in the busyness. But the point here is there are a remarkable range, as I hinted before, at the anti-inflammatory effects of CBD. And just to highlight it, just look at number 3, reducing the amount of pro-inflammatory cytokines, IL-1 beta, IL-3, 6, 12, 17, TNF-alpha, and IFN beta gamma. And it also increases some of the anti-inflammatory cytokines. And that is just one of 8 of its anti-inflammatory effects. So I do think for inflammatory disorders, whether they're in the gastrointestinal system, skin, nervous system, that the anti-inflammatory effects are quite potent and interesting and diverse. And so CBD really may have an important role in some autoimmune and inflammatory disorders. This slide summarizes some of the effects of cannabinoids in preclinical, so in animal studies of epilepsy models. And I'll focus your attention on the 2 histograms on the right side here. These look at delta-9 THC drugs or drugs that act just like delta-9 THC. And on the far right, CBD and cannabidivarin or CBDV, which is another compound that's being used to study the effects and very similar in its profile to CBD. So what you see here is the green portion of this histogram bar is anticonvulsant. So THC, it's about 65% in the animal models, and these are up to 25 different animal models across multiple species of epilepsy, that it's anticonvulsant or anti-seizure. There's a drop of proconvulsant, a drop of mixed, where those can be either pro or anti, and then about 30-35% where it has no significant effect. By contrast, for CBD and CBDV, about 75% of the studies show it is anticonvulsant, and about 25% show it has no significant effect on seizure threshold. But CBD also has no proconvulsant effects in any of the models studied to date. This slide summarizes research that was began by Salantiev's group and published in 2013, and more recently by Evan Rosenberg, who's a colleague, was at NYU, where he did his MD PhD under Dick Chen and myself, but he's now a neurology resident at Penn. So what Salantev found was that there's an endogenous, that means a compound made by our brain, LPI, it's a lipid, lysophosphatidylcholine inositol, and LPI binds, this is the GPR55 receptor, and what it does is, what Salantev found is it will increase. So when LPI binds the GPR55, it increases excitation. What Evan Rosenberg found was that when when LPI binds to the GPR55 receptor, it decreases inhibition. So what you have here is LPI is a perfect nightmare for epilepsy. It increases excitation and it decreases inhibition. Epilepsy can be considered one of the disorders of excitation to inhibition ratio being thrown out of whack, and you have too much excitation and too little inhibition, and LPI makes both of those worse. It's even worse than that. After a seizure, LPI, which can be generated from LPI, the seizure itself will lead to increased levels of GPR55 on nerve cells and therefore create a self-perpetuating cycle. CBD blocks the effects of LPI both on the excitatory glutamatergic neurons and on the inhibitory GABAergic interneurons. So again, CBD, in the cases where GPR55 and LPI are a positive feedback loop, and this is probably some of the cases where we just see truly miraculous effects from CBD, because if this loop is driving someone's epilepsy, then CBD is the way out of the loop. So let's touch base on some of the pharmacology of CBD. This is from an article published several years ago, sponsored by GW, looking at the Epidiolex, which is 99% CBD in sesame oil. So as I think everybody knows, all of the cannabinoids, CBD, THC, and essentially the others, are all fat soluble. And that carries some important caveats with it. Oral absorption is poor or terrible. So if you were to take CBD as a capsule, as an oil, and took it by mouth or put it under your tongue, and if you put it under your tongue, most of it's going to end up in your stomach anyway. But you will absorb, if you take it on an empty stomach or after a vegetarian or high carbohydrate meal, you'll absorb probably 4, 5, or 6%. A really, really tiny amount will actually get in to your blood and have a chance to make it to the liver, where there's already fairly significant first pass metabolism. If you consume CBD or THC, they're both very similar as fat soluble compounds, you can get up to 20% absorption, so 4 to 5-fold. And again, for people who are paying for this, and it's expensive, as many of my patients do, and people use it off-label, they're getting it from places where, a recessed soda in New York, which I think is a homeopathic dose, it costs $5. It's a seltzer basically with CBD, and it has 10 milligrams of CBD. Keep in mind, in the trials I'm telling you about, we gave children over 1,000 milligrams very commonly. So you're talking about 1/100th the dose, and only often 4%, 5% is getting absorbed. Another important point in understanding the pharmacology of cannabidiol or THC is that the absorption and first pass metabolism vary tremendously between and within individuals. So in this table from the paper by Taylor, what I'll point to here is these numbers in parentheses. This is a 1,500 milligram dose given to normal volunteers, and they're looking at the Cmax, what the maximum level is in the blood. And don't worry about the 292, which is the actual nanograms per milliliter, but look at the number on the right, 87.9. That's the coefficient of variation, and that's enormous. For many drugs like Tylenol or Advil, acetaminophen, or ibuprofen, you're looking at coefficients of variability of 10%, 15%, because they're water soluble and they're so much better tolerated. And again, the area under the curve, which is how much of the total drug gets absorbed, again, you're looking at 78%, which is still just an enormous coefficient of variability. So these are relatively dirty drugs, not in a bad way, but just that there's so much variability within and between people that it's not nearly as consistent as we are used to in prescribing medications. So when I got into CBD, and I would say this is still very much still there, there's a lot of religion. That is, people come into it with very strong beliefs. People would bring their children to me, having had bad epilepsy for 6 months, saying, I didn't want to use a pharmaceutical product, but I want to use CBD because it's natural and I hear it's great. And a couple things about that. One is that it is natural. It does come from a plant, but that doesn't mean anything. There's another compound that comes from a plant grown in the highlands of the Andes Mountains that's been consumed by humans for thousands of years. And if you chew the leaves, it seems to be fairly benign. But when you take 100 pounds of those leaves and you distill out a quarter of a pound of a white powder called cocaine, it is a very, very dangerous bad drug. It's natural. It's as natural as CBD is, but it's just not a good drug, and it's not a safe drug. I think CBD is much safer than cocaine, but keep in mind, it is a drug. We're not consuming a plant. We're not doing as native populations have probably done for long periods of time. We're taking this to a chemical plant and doing extractions, which involve often chemicals to extract it and concentrate it. So when we look at the trying to understand how well this works, in medicine, it's not that randomized controlled trials are the best way to get a definite answer. They are the only way to get close to the truth. And of course, randomized controlled trials can differ depending on populations, randomization protocols, many other factors. So they're very far from perfect. And I'll get into some of those features, but they're so much better than either open label studies or studies where the investigator or the patient's single-blinded, where someone knows what's going on. So in a double-blinded randomized controlled trial, as we did for CBD in these pediatric epilepsies, neither we as clinicians nor the patients or parents knew who was getting the active drug and who was getting a placebo. And this assignment was done at random. None of us knew who was in which group or how it was done, other than, patient number 3 at NYU would be put into one group, and we just didn't know. It helps limit bias because subconscious bias, we're all biased. Doctors are, scientists are, patients are, families. One thing I found out in doing this research, which came to me as unintuitive and a very big surprise, is that the placebo response in pediatric trials is roughly twice as great as it is in adult trials. Now that seems crazy to me in that parents are just reporting something like seizures, what they see in their kid. It's not that they're reporting themselves, but because parents are so emotionally invested in their children, probably more than they are in themselves, they tend to be biased even more so. And then finally, another wonderful part of randomized controlled trials is we get to see what the adverse effects that are related to study drug. There are some studies where the placebo group has 80% side effects. And so that tells you that that's a very high number, but you need to consider that, because if the active treatment group has 78%, then really it essentially doesn't differ, assuming the severity and other factors are the same from the active drug. So this is the first study that was published now 4 years ago in Dravet Syndrome. It's from a group of a number of epilepsy centers, both in the United States, England, and Europe. And it was a phase 3 trial of Epidiolex in children 2 to 18 years of age with Dravet Syndrome, which is a very severe treatment-resistant epilepsy syndrome. We only used one dose of CBD, 20 milligrams per kilogram. So again, the average adult, we hope, is 60 kilograms. Unfortunately, with the obesity epidemic, that's no longer true. But that would translate to about 1,200 milligrams per day for the average 60 kilogram adult. And the mean age was 10 years old. The median seizure frequency of countable convulsive seizures was 13 per month. Most of these patients had been on many drugs in the past and averaged 3 drugs to which CBD was added. Serious adverse effects were reported in 3 in the CBD group versus 3 in the placebo, so about 3-fold higher. And discontinuation, that is, the parents wanted their child to leave the study because of side effects, occurred 8 times in the CBD group and 1 in the placebo. Again, just pointing out that CBD is a real drug. Now, if you look at the graph, what you can see statistically, less than 0.01, that CBD did work better than placebo. That's the median percent reduction in convulsive seizures. You can see it's about 39% for CBD and about 13% for placebo. So about a 26% differentiation. So again, one caveat here is that CBD is not a miraculous drug. Here we are adding it to 3 other drugs, and we got an average of a 26% reduction in the convulsive seizures, which was meaningful. And for some of these children who had a much greater reduction, it was extraordinarily meaningful to them. But many had a minimal benefit, and some actually had no benefit or slight worsening. So keep in mind, this is not a panacea. This is not a miracle drug. It clearly worked. Could not be happier. We put a lot of time and effort. And when the blind was unmasked, I remember getting it. I was on vacation as well, and because of all the brouhaha about keeping it secret and potentially people trading on the stock, I had to go to a little corner of the conference room and had a very big smile on my face when I opened the slide and saw that we got statistical significance. So these were the next 2 studies that were done. The first one, which was very similar to the one I just went over for Dravet, it was in Lennox-Gastaut Syndrome, another severe pediatric and adult epilepsy syndrome. So here the patients were ages 2 to 55 years of age. And on the left side, you can see the placebo had about 21%. So again, a significantly higher rate of placebo, but fortunately the efficacy was also higher, about 44% in the CBD group, and it again reached statistical significance. And the study on the right was one that I led and was published also in 2018, where we looked at two doses now. One was the 10 milligram per kilogram per day, and the other was 20. And what you can see, not surprisingly, is the 20 worked a bit better than the 10, but also not surprisingly, the side effects were significantly lower in the 10 milligram per kilogram per day dose. So that's the one that's now being recommended as the target dose. People can go higher, but often 10 milligrams per kilogram per day is a bit of a sweet spot, I'd say, where you get 90% of the efficacy, but many fewer side effects. And another thing I'll point out, which is also true for the Dravet study, choosing primary outcome endpoints is really critical. We pondered this long and hard. Most epilepsy trials have used the 50% responder rate, which means if there are 100 people in the trial and X number have a reduction in their seizures of 50% or more, those are considered the responders. And you look at the responders in the treatment versus the placebo group. If we had done that with this, CBD would not be approved by the FDA because it would not have reached the statistical significant milestone, or it would have barely made it over the hump. And so we chose this outcome because this has been seen in other studies, for example, in Lennox-Gastaut with clobazam, and we use that as an important lesson, and I'm very happy we did. What did we learn about side effects from these randomized controlled trials? Well, like I said, CBD is a drug and we're giving it at pharmaceutical doses, not the natural doses that would be consumed by someone. And the main groups are CNS, somnolence and sedation, gastrointestinal, decreased appetite and diarrhea, elevated liver function enzymes or transaminases, increased rates of infections, primarily pneumonia, and then drug interactions because CBD inhibits the liver enzyme, the cytochrome 2C19. And this is most important for drugs like clobazam because clobazam has an active metabolite, N-desmethylclobazam, and this will raise those levels significantly. As I said, the withdrawals were about 7 or 8-fold higher in the CBD group, mainly due to elevated liver function tests and due to somnolence. So there have been some trials of CBD and CBDV in other epilepsies. One study looked at focal epilepsy using a synthetic CBD applied by a transdermal route. This is Zynerba's study, and it was open label, but there was no evidence of efficacy versus placebo. There was another study led by GW using CBDV, which, as I remind you, is very, very similar in its receptor profile to CBD, and again, did not differentiate from placebo. I should mention here, I don't think I have the slide, but there was one further study that Elizabeth Thiel led as well on Tuberous Sclerosis Complex, which is a genetic epilepsy, mainly with focal seizures. And in that study, CBD at very high doses, they looked at 25 and 50 milligrams per kilogram per day, which are absolutely stunningly high doses. For me, at my weight, I think that would be well on the 50 milligram per kilogram per day dose, getting up to 4,000 milligrams per day. And that's what many people consume in a month or two for different disorders. So really high doses, but it did show efficacy in tuberous sclerosis complex. And I just want to briefly touch base on autism, because I think for those in the pediatric field and those who follow, there's a lot of interest for CBD in autism and a lot of open label data, but not a lot of controlled data to date. This is one study in which 20 patients had a transdermal synthetic CBD, mainly with Fragile X. It was open label. They looked at a scale that looked at anxiety, depression, and mood, and they found that subscores did quite better with the CBD. So this was a very positive study and led Zynerba to go on with a larger trial that's now ongoing with CBD and Fragile X, looking at behavioral aspects. There were, again, similar rates of side effects to what we saw. Most were mild, none were serious, and they focused on the GI system. They also found urinary tract infections more frequent in the study as well. This is another study, which I believe is from Israel, looking at a combination of CBD and THC, which is a commonly used mixture. It's interesting, just to touch base on that for a second. So THC, remember, is psychoactive. It stimulates the CB1 receptor. The CBD is actually a little bit of an antagonist to this. So it doesn't stimulate the CB1 receptor like THC. If anything, it slightly reduces the effect of THC on that receptor. And I apologize, it should be 20 parts CBD to 1 part THC, so that's reversed. And this was primarily in children, but went up to age 22. And essentially, they found an open label retrospective review, self-injury and rage went down in about 68%, hyperactivity went down in 68%, sleep problems were improved in 71%, and anxiety was reduced in 47%. But keep in mind, for some, sleep problems got worse in 45%, anxiety got worse in 23%. So again, many of us have different forms of these receptors and have different metabolic pathways. And so the drug that can be beneficial for one person can be negative for another person. And that's true for most drugs that we use in medicine. This is an additional study, I think again from Israel, a CBD-rich cannabis solution or liquid with oil. 60 children with autism were in there, and 60% were much improved or more so. Again, side effects, sleep disturbance, irritability, issues with appetite. One child, if they didn't do well with the high CBD, they got a more enriched THC preparation, and one of those children developed a psychotic issue. And then another study also looked at the 20-to-1 CBD to THC, and again, very similar, significant improvement in 30%, moderate improvement in 54%. In that study, it was only 9% had no benefit at all, and none were worse. Side effects were pretty mild and limited. The high, again, keep in mind, some of these children were receiving THC, and some were given relatively higher amounts if they didn't respond to the initial enriched CBD preparation. And there are several studies ongoing now in individuals with autism. Javier Castellanos, who's a colleague of mine at NYU, is doing one with CBD through the GW Pharma using Epidiolex. And there's another one that I'm involved with Eric Hollander from Montefiore on CBDV, and that is also a GW preparation, but not CBD, but CBDV. So these studies are ongoing. These are, I think the first one is open label, the second one is blinded, so that may give us some of the best early indications of the true efficacy, at least of CBDV, in treating individuals with autism. But the anecdotal reports are that it can improve negative behaviors like irritability and aggression, can lessen anxiety, and help improve sleep in some of these children and young adults. The next topic I want to touch on, which is also relevant for children, but also adults, is anxiety. The anxiolytics that are out on the market have issues. So benzodiazepines are very commonly used. Those are the Xanax or alprazolam, clonazepam, diazepam. There are many of them. I'm not sure the situation in Canada, but in the United States, I would say they're horribly overused by physicians. My wife, for example, had tinnitus after having a benign meningioma taken out. She went to an ear, nose, throat doctor who gave her 90 clonazepams. Why he gave her clonazepam and why he gave her 90 is a mystery to me. But she came home from the pharmacy with 90 Klonopins. And I would say benzodiazepines together with opiates are the two most addictive categories of medicine that are available for people. There are others that get close, some of the stimulants, but these drugs are just so habit forming, and nobody knows who's who. I mean, doctors get addicted, nurses get addicted, and keep in mind, in the US, where we have a terrible issue with overdoses, I think you can see here on the point over here, overdose deaths, about 100,000 of opiates. I think 20% of those are on benzos at the same time. So it's a major, major issue. The other drugs that are out there are the serotonin reuptake inhibitors and the serotonin norepinephrine reuptake inhibitors, which again can be very effective, but they have their own negative effects. I think buspirone is one of the more benign ones, but not nearly as effective. And as I said, the benzodiazepines really have a lot of issues in both being addictive, and also you can see one benzo script increased in the United States from 28 million in 2003 to 63 million in 2015. So what's changed in the United States that the number of these scripts has gone up more than twofold? The number of deaths from benzodiazepines where they were on board and an overdose went from 0.58 per 100,000 to 3.1 over a 14-year period. So again, up almost sixfold for that. And as I kind of hinted at, there were about 11,500 deaths in the United States in which benzodiazepines contributed. So these are nasty drugs, and I would just recommend people give them out relatively without much warning to the patients who get them, and then they're rarely indicated for chronic use, but they are used that way by psychiatrists and even internists and neurologists all the time. The CBD-induced anxiolytic effects are probably mediated by the serotonin 1A receptor, although that's not 100% proven at this point. And then this looks at some of the preclinical work on a chronic stress model. And this is from Campos about almost a decade ago, but you can see here in the CBDV group, a significant benefit in the stressed condition, especially here on the right side versus the non-stress. So again, there's multiple studies on these anxiolytic effects in preclinical animal models. And then there's also studies dating back a decade in humans. Individuals with anxiety disorder or without are asked to do a public speaking, which typically will increase heart rate and levels of anxiety. And you can see the placebo group in red, the CBD group here, and healthy controls who don't have any social anxiety disorder. And again, the sedation, interestingly, goes again with CBD in between. So again, there's evidence from both animals and humans that CBD does have anti-anxiety properties. It has not been rigorously tested in randomized placebo-controlled trials of patients with anxiety disorder, but hopefully those will come out soon, because I truly believe the drugs used to treat anxiety have many, many side effects. Clearly the benzos in my mind are the worst, but the serotonin and serotonin norepinephrine reuptake inhibitors can have sexual dysfunction, can cause weight gain, can cause GI side effects. So again, for many people, they're really not well tolerated. So I want to finish up on what I like to refer to as the Wild West. It is a phenomenal social event that has occurred in our lifetime in medicine. Imagine there was a drug for breast or prostate cancer, and it came from, let's just say, the coca leaf, and you could buy cocaine, and it was reportedly beneficial to treat either breast or prostate cancer. Now, number one, if that indeed was identified and there were good anecdotal observations, what we traditionally would do in our medical system would be to do large randomized controlled trials, whether they're funded by a drug company or whether they're funded by the NIH or equivalent organizations in other countries. I'm agnostic, but the goal would be to do large randomized placebo-controlled studies. And my goodness, if cocaine could cure someone of breast cancer or prostate cancer who had, stage 4 metastatic disease, despite the significant negative effects of cocaine, I would argue anyone with those disorders should get access to it as long as the benefits outweigh the risk. And that's a risk-benefit equation that needs to be informed by science. But imagine what would happen if 5 women with breast cancer and 5 men with prostate cancer went in front of a state legislature and said, this drug cured our cancer, you have to approve it. Now, mind you, Steve McQueen did that at government hearings to say that Laetrile helped cure his cancer. It was later, he died not long after giving that testimony, and subsequent large double-blind placebo-controlled trials, one of which was published in the New England Journal of Medicine, showed that Laetrile not only did no benefit to people with cancer, but actually caused more harm, because I believe it's from the apricot pit and contains some arsenic in it, which is a toxic compound, as you know. So again, you can't trust people like me, doctors and scientists. You can't trust patients because we're all biased. So the idea that a group of parents or adults who have disorders went to the legislature to say you need to approve medical marijuana, I think is a dangerous precedent. I'm not against it because I do think, as in epilepsy cases, if you have a child with uncontrolled seizures and you've tried 10 or 12 or 14 available medicines and had surgery and had neuromodulation with a vagus nerve stimulator and had a ketogenic diet, and if your child failed all that, well, my goodness, for that child, probably it's a reasonable thing to allow it to be used. But it should be used in a supervised way. And simultaneously, we should be pushing hard as a medical and governmental society to get the answer. Does it help these kids or not? If it helps them, my goodness, they shouldn't have to spend $300 a month to get the compound from a mail order place in Colorado or California. It should be provided by their insurance company at no charge or minimal charge. So the idea that people will go to a legislatures, I think, are poorly equipped. And people like Senator McGovern in the United States, I think, may have literally contributed to the deaths of millions of people when they heard testimony and convicted saturated fat causing heart disease and told Americans to increase carbohydrates, and not directly, but the implication was that things like sugar were safe, white flour, white rice were safe, eat as much as you want, but avoid saturated fat. The result is an obesity epidemic, a diabetes epidemic, a non-alcoholic fatty liver disease epidemic. Cancer is driven by these same processes. So horrible things were done by smart people meaning well, but not being informed by rigorous science. And that's where we are here. So with medical cannabis, religion is belief without evidence. If you hold whatever religion you may believe and you're a devout worshiper, that's fine. If you want to believe in this prophet or that prophet or this set of beliefs, I think everyone's entitled to that. And you don't need any evidence. But if you want to go into science or medicine, you need to have doubt despite 99% certainty, because everything we know in science today will be found to be partially, largely, or completely wrong 10, 30, or 100 years from now. So in America, and again, I don't know the situation in Canada, but you literally can have patients of mine going into a dispensary in New York City, and the staff there may have a high school diploma, and they will be influencing, no, Dr. Devinsky said use high CBD, but I really find that you need some THC if you're going to get some benefit here. And again, they may end up being right, but without any knowledge, it's a very dangerous way to have medicines prescribed. Professional and lay individuals often believe they have answers. I was once in a meeting with a man who worked at a large company and was influencing a lot of their decisions, and he was saying, I've counseled thousands of people with epilepsy about how to use the different ratios and strains and mix things up and move them. And I'm like, oh, interesting, and I'm just listening. I didn't want to get into a confrontation. As I'm in the meeting with this gentleman, he got called. His child, who had epilepsy, was being admitted to the ICU with an uncontrolled seizure lasting more than 30 minutes. So here's a man claiming he had it all figured out, and yet his child's going into the ICU. And I'm thinking, what's wrong with this situation? And as I gave with the McGovern hearings, beware of the law of unintended consequences. We often think, for example, and I've heard many people say this, well, it's natural, so it's not going to do any harm. No, that's wrong. There are people who have had serious adverse effects from CBD and THC, not commonly and dependent on the dose and their health, but it definitely happens. And once you believe in something, no matter who you are, scientists are just as much to praise as others. There's confirmation bias. We see everything that supports us and find reasons to affirm it, and everything that refutes us and find reasons why it's wrong or just simply ignore it. And then there's a sunk cost bias. Once I believe something, and especially if I'm a thought leader and my ego is now tied to it, my reputation is now tied to it, I will do everything in my power to make sure my viewpoint is not overturned. And that's a terrible thing in science and medicine. So in conclusion, just to wrap up and go over the highlights, CBD and THC, there's a lot of data that they are both anti-seizure drugs in animal models. There's excellent randomized placebo-controlled trial data that CBD reduces convulsive and drop seizures in Dravet Syndrome, Lennox-Gastaut Syndrome, and Tuberous Sclerosis Complex. There's no current evidence that CBD or CBDV is efficacious for focal epilepsy, except in the specific situation of tuberous sclerosis. We need data on other epilepsies, the generalized genetic or idiopathic generalized epilepsies and many other rare epilepsies. There's some signals of efficacy, but we need to have good studies. We have almost no data on THC in epilepsy, either for its safety and definitely not for its efficacy. And of course, there's a concern. Many parents are giving young children, babies, THC, and that's dangerous. I just had a call with a friend of the family's friend who has a child with Tourette syndrome. And at the end of the conversation, the dad, who's an intelligent man, said, well, I am microdosing him with psilocybin. So again, this is where we are as a society. He may be doing more good than harm. He may be doing more harm than good, but it's a dangerous road to traverse. And then we need a lot more data on the lay consumption of these compounds, especially in special groups, such as pregnant women who could hurt their fetus, as well as young children. So I'm going to stop there. Thank you again for this kind invitation, and I will open it up for questions.
Thank you very much, Dr. Devinsky, both for an excellent talk and for also leading by example for doing these kinds of trials that we do need to sort out what is evidence and what is hyperbole. So I really want to thank you for a very informative lecture. Alan, do we have some material that's been entered?
So there aren't any questions yet. Oh wait, one has just come in. If you do have any questions, please type them into the Q&A box. This question from Roman. What doses may have been used in the autism trials?
So the doses range quite widely. Some of them start with relatively low doses, I would say in the range of 2 to 5 milligrams per kilogram, but some have gone up to the 10 to 20 milligram per kilogram range for the CBD component. And most of those, the THC was about 1/20th the dose, although as I showed some of those trials, if the children didn't respond, they then used higher levels of THC for those children. So, but I think 10 milligrams per day is kind of towards an upper range to give a child THC, although that effect is going to be muted by the co-administration of the CBD, which is going to partially antagonize the CB1 receptor.
Great. Thank you. There is another one in the chat question. Is there a known pharmacological difference in makeup between sativa and indica strains?
Yeah, so traditionally the sativa Chinese strains were richer in THC and relatively poor in CBD, whereas the indica strains from India were relatively high in CBD and low in THC. Those species have been so interbred, that would be like trying to find, someone of European white descent and say, oh, I could tell by your nose and your eyes that you probably come from Slovenia or outside of Normandy, France. And there's no way to do that anymore. The amount of mixing that has gone in those strains is enormous. But in dispensaries, they're often labeled. So anything that's high CBD is called an indica, and anything that's high THC is called a sativa, although the actual plant that it's derived from may have nothing to do with those labels, because they can get rebred and bred in different ways to have high CBD or high THC. So you can take an indica, and which should be high CBD, and you could breed it so it's very high THC, and it still may be largely genetically an indica species. So again, huge amounts of mixing.
Great. Another question from Linda. There are products that are nano-emulsified. These products are supposedly more bioavailable. They also avoid first pass issue as they supposedly are absorbed in stomach. Are you familiar with any studies utilizing nano-emulsified cannabis products?
I'm aware of people using them. I am not aware of published pharmacokinetic data. So actually, if someone has that, I would love, my email is, you can just Google me, but I'd love to see those studies. Again, the idea that the stomach is not the best place to absorb things because of the environment. But again, depending on what the nanoparticles are made of and what's in there, they might get absorbed from the stomach, but I think still some of the stomach ends up in the portal circulation and does drain into the liver because, again, if we eat a toxic mushroom and it's got a horrible, dangerous compound in it, you want that compound to go through the liver before it goes to your brain. And biology is good at working those things out. So I'm not, that sounds like something that's been promoted by someone selling it. I would love to see any data supporting either of those claims, either that it skips the first pass effect, either that it's better absorbed, or that it's absorbed in the stomach. So I'll be skeptical of all three, but I have no doubt they could all be true.
A question in the chat from Margot. When do you check liver testing in patients on CBD?
So I think it's important to do it. Again, depends on the dose. If people are giving what I would call closer to homeopathic doses of like 20 milligrams a day or 30 milligrams a day, I think the chance of liver injury is really quite tiny, and I'm not sure you ultimately need to check it. For example, that recess soda that I mentioned has 10 milligrams of CBD in a can. If you're drinking 2 of those a day, I don't think you need to worry about your liver test because it's nothing. A glass of wine is probably going to have more effect on your liver than that amount of CBD. But if you're using it medicinally and you're taking a dose, let's just say as an adult like you, 600 milligrams a day, and you're taking it on a regular basis, then it's probably worth checking a liver function test, 3 to 8 weeks after starting it. I think if the liver enzymes are unchanged, you don't need to check it again. But the other situation where you should check it more frequently would be if someone has known elevated liver function tests before they went on the CBD, or if they're on another compound like we found in our studies, valproic acid, Depakote in the US, which is known to be hepatotoxic. Those individuals had much higher issues of significant liver function test abnormalities. So if you go in with high liver function tests before you use CBD, if you're on another drug and you use high doses, those 3 features make you quite likely to have an issue with liver problems.
A follow-up to that, I guess, would be from someone in the Q&A portal. Is the evidence conclusive on what is a safe dose of CBD?
I don't think we can say it's conclusive because everybody's different. And, I can have 4 cups of coffee a day and, feel slightly energized, but no side effects. I have a relative who could have half a cup, and he's an adult, my age roughly, half a cup a day, and he feels terrible. He just can't tolerate it. So I think there's a huge amount of individual variability, but I do think, to be candid, most people tolerate up to 300 to 600 milligrams a day without any significant side effects or problems.
Another question in the Q&A portal. What would you consider to be the most important next steps with regard to furthering research in the field of therapeutic CBD use for pediatric seizure disorders?
So I think we need more data on the generalized epilepsies and focal epilepsies, which together probably represent 80% or more of all the pediatric epilepsies. I think because many parents and physicians are interested, I think there should be more rigorous trials of THC, whether it's used alone or with CBD. But I think both of those are areas where we need randomized controlled trials.
One more question here, maybe 2 more. I may have missed this as I joined late, but what does the evidence say about CBD and liver injury and somnolence? Is this caused by the CBD or drug-drug interactions between the CBD and other drugs, NHPs? So
it's a good question, and the answer is both. So CBD all by itself, there's no doubt can cause liver toxicity. It can also cause somnolence by itself. A lot of the children in both the Dravet and Lennox-Gastaut, and I think less so in the tuberous sclerosis studies, were on a drug called clobazam. And one of the interactions that CBD has, as I mentioned, it inhibits cytochrome 2C19. And because of that, the levels of desmethyl clobazam will go up to 400 to 500%, huge increases, 4 to 5-fold. And that is an active metabolite. It does cause sedation. And there is a correlation between levels of N-desmethyl clobazam and sedation in these trials. So I think the sedation from CBD is twofold. Partly it's direct from CBD itself on the brain, and the other is the effect on the liver to increase levels of other medications like endesmethylclobazam.
A question from Roman. Do you suspect that the analgesic effects of CBD may be related to the neurologic effects or the anti-inflammatory effects?
I think probably both, if I had to guess. I think, number one, the analgesic effects of CBD remain to be fully tested, but I think there's evidence it has some. And I think it's going to probably be, if I had to use my guesstimation, both the TRPV receptors, which are the capsaicin activated receptors, so that probably can help block some of the TRPV pain. And in addition, it probably also does, through its anti-inflammatory effects. Pain is often associated with musculoskeletal injuries where there's inflammation. So anything that will take down the inflammation will often take down pain as well. So I would, my clinical gut is that probably both mechanisms may be relevant.
Is there any evidence that CBD could impair driving if it, for example, induces somnolence?
I think that's possible. I think, again, I went to a party once that was billed as a CBD party. And one of the people I went with actually had a cocktail when they got there. It was a nice summer afternoon. So I think there was vodka or whatever was in the cocktail, and she had a fair amount of CBD, and she fell asleep on the car ride home. So again, you can't separate the alcohol from the CBD, but I think, combining things like that can make you even more tired. So I think you should know yourself, and it is not psychoactive in that it doesn't produce a high, but there's no doubt it can produce tiredness. And if you're on other medications like alcohol or diphenhydramine that cause tiredness, that effect may be magnified. But in general, at most of the doses used, I think people are safe to drive, although like everything, there probably are some occasional exceptions.
Thank you very much. I think those are all the questions that are in the chat right now. There's a few more. Do we have time for a few more questions?
I think so.
Okay. Do you know of any research looking at the long-term effects of CBD, THC on receptor neuron firing and how it might impact behavior in the long term beyond just desired effects like reducing anxiety, possible psychosis, etc.?
It's a good question. As I said, we know the effects of the THC on the CB1 receptor and CB2 receptor outside of the brain. Those have been studied for a long time. CBD is a much, much more complicated compound. It literally influences many, many, many receptors. So it's more pleiotropic in that way. And the question, I think, gets at a really important thing is, what are the long-term effects? And the answer is, we don't know that for almost any of our drugs. Any drug, whether you give a dopamine block, a serotonin reuptake inhibitor, those can cause very long-term changes in those neurons, desensitization, additional receptors, modified receptors. And because almost every drug that gets approved, at least in the United States, is subjected to a 3-month, maybe a 14-week trial, long-term effects are just not looked at. And that was, of course, one of the issues with benzodiazepines. You give someone with anxiety disorder clonazepam, it's a miracle drug. It's absolutely amazing for 2 weeks. And then all of a sudden the person needs more. And if you look at those people a year later, the evidence is that they're more likely to be more anxious. And now you put them on a roller coaster where for 4 hours a day, they feel better, they take more clonazepam, they feel worse, and then they take more, they feel better, and they're just up and down, and you kind of created a little bit of a junkie. And I think overall, their baseline anxiety levels tend to be worse. So short-term and long-term are very different, but sadly, medicine has focused mainly on relatively short-term periods.
And a final question, would people taking CBD and other cannabinoids test positive for cannabis screening tests, mainly targeting THC?
The answer should be no, with the caveat being, depending on where you get the product and how well purified it is. So you can get a product from a dispensary or from a website that says it's 100% CBD. And if there was 3% THC in there, I wouldn't be shocked from some places because this is not being well regulated. There are not regular testing of all the sites that you could buy in America anyway. CBD online. There's hundreds of outlets to buy CBD. And so if one of them contains small amounts of THC, it could well, some of those tests are quite sensitive. And cannabinoids, because they are fat soluble, they do accumulate over time. So if you're taking the same CBD solution and it's got 5% THC in it, then yeah, quite likely you could end up having a positive THC. Although I would hope that that's an infrequent occurrence. I would imagine it has occurred.
Thank you, Dr. Devinsky. Jason, I'll pass it back to you or to James. Final thoughts?
Yeah, I mean, this has been a fascinating talk. I think you've hit a number of topics that are of great interest. As you can see, we've been taking questions right up until now. I think you've really highlighted the incredible importance of doing the right studies, doing the randomized controlled trials, making sure that policy is being driven by evidence as opposed to anecdote and marketing. And we're in a very similar situation as you in the South. So we're very excited about the work you've been doing, about the work you continue to do. And thanks again for taking time out of your vacation to join us today to provide this lecture.
My pleasure. Thank you all. Be well.